So this next recording, we're going to continue our investigation of biomacromolecules, but now we're going to look at protein. And I'm probably going to spend the most time on protein of all the biomacromolecules at this stage in the game, because proteins are really, really important. They're all very important, but my background is in proteins, and so I'm going to share more information with you on proteins. Now remember, we've got 4 classes of biomacromolecules: carbohydrates, lipids, proteins. And proteins are a polymer composed of amino acids. Again, when we think of proteins, we can depict them different ways. They're these relatively large globular molecules. You think of cheese, you think of steak, you think of fish, you think of flesh. Like, this is protein. Because the muscle in animals is made of these long protein fibers. That's what allows us to move. And so it's rich in protein. Right? Now, proteins account for more than 50% of the dry weight of most cells. There's a lot of proteins going on there. Now, again, most of the weight of a living thing is water, so don't think that there's that much protein, but of the components of a cell, proteins are important. They can be structural, they can serve as a role in communications between cells, within a cell, they can be effective components of the immune system, but what I want you to remember is that proteins do stuff. They're going to be the enzymes that are going to direct the biochemistry of the cell. And so everything that we look at, when we look at how DNA works, how your metabolism works, how cells reproduce, ultimately proteins are the things that are doing things, right? And so you have this short list here that you can see about enzymatic proteins: structural, storage, transport, hormones, receptors, motor proteins, defensive proteins. Proteins do stuff. The idea of the enzyme is very important, and to understand the central role of proteins in the cell, we have to understand what an enzyme is. And we'll come back and review this again later in the quarter, but for now let me introduce it because I'm going to speak with this language and I want you to understand it. So enzymes are catalysts, right? So again, from your intro chemistry, you should understand the basic idea of a catalyst. It's something that participates in a reaction, promoting that reaction, but is not used up by that reaction. So again, when we think about efficiency in life, if I can create a molecule that can participate and do it again and again and again and again, and I don't have to build it every time, it's much more efficient than having a one-shot per molecule kind of process, right? So if we think about an enzyme, in this case sucrase, is going to take a substance, sucrose, and break it down into its 2 component monosaccharides, glucose and fructose. And it does that by catalyzing that reaction. And we'll talk again about those details later. But again, we have our enzyme, and it binds to its substrate and then produces the products. All right. Now, proteins are polypeptides. They are many monomers put together. So a poly, many peptides. The peptide bond is the bond that you connect amino acids with. All right. Protein polypeptide can be used relatively synonymously. There are subtleties to the use of language when we talk about protein chemistry, but if you use those 2 synonymously, I wouldn't blink. All right. So amino acids now are again our monomer that is we're going to connect together to make our polymers. When we look at each individual amino acid, we have a general structure that can be seen. And that general structure is an amine group on one side, a carboxyl group on the other, linked by essential what we call the alpha carbon. And then we have the R group. Okay, so R is not an element there. It stands for a variable, right? So the side group then is standardized. There are 20 different R groups that make up 20 different amino acids. And this then is a chart that shows you all those amino acids. Now we can basically break those down into 2 important categories. Nonpolar amino acids. Nonpolar is going to be hydrophobic, right? And when you look at the R groups highlighted in white here, they're all hydrocarbons, in essence. So methionine, you have that sulfur in there, but in essence, these are highly nonpolar bonds, and hence these side groups are going to not want to interact with water. They're going to be hydrophobic. They're nonpolar. Versus the charged and polar groups are going to want to interact with water. Right? So these 2 fundamental groups of amino acids, when we put them together in a chain and into an aqueous environment, are going to have different effects. And that's going to be one of the fundamental concepts of protein folding, which we'll get to in just a couple lectures. Now, again, amino acids linked together form polypeptides. the bond that links them is called a peptide bond, and hence the source of the term polypeptide. So when we look at that, again, we see a dehydration synthesis reaction linking here one amino acid to a growing amino acid chain, and that bond between the carbon and then the nitrogen is called a peptide bond. Right? So that's the fundamental introduction to the idea of proteins as polymers. Next, we'll look at protein structure.